Hi everybody, in this video we are going to discuss about the subject called as the AIDS, okay, the topic called as the AIDS, and in this video we are going to discuss about its mode of infection, okay, mode of infection. Now when we look at the virus, okay, causing AIDS, okay, we know that we studied in the previous video that AIDS is caused by a virus called as HIV, human immunodeficiency virus, Now we will discuss the mode of infection, how this disease is caused, okay, how this disease is caused. Now we already said another point, that is this HIV virus is a retrovirus. It belongs to retrovirus, okay, it belongs to retrovirus, okay, please remember this. Now let us see. Now if this is the virus, okay, this is the virus, it is called as what? The retrovirus. This retrovirus, which is with what? Viral protein code, okay, it is there with the viral protein code and also the viral RNA code, okay, this is the viral RNA core, means RNA is present, okay, RNA is present in a core like structure. Now here this particular virus, okay, HIV virus belonging to retrovirus group. Now first, how it attacks, step by step we are going to see now. Now this retrovirus infects the normal cells, Now normal cells means what? Healthy cells, okay, normal cells means healthy cells. Now question here, there is a one question, like which cells? Normal cells means there are different cells, there are brain cells, we say, there are muscle cells, there are skin cells, there are bone cells, like that there are different types of cells in our body. But please remember here, when we are talking about the normal cells, it affects or infects, okay, it infects the cells which are called as the blood cells, called as the macrophages, okay, called as the macrophages, which are playing a crucial role in immune system, okay, in immune system. So these are cells, okay, macrophages are cells which play a very important role in the immune system, okay, in the immune system to protect our body from various disease causing microbes. So these are called as, now those normal cells are called as what? Macrophages, okay, normal cells are called as the macrophages. And remember, in the very beginning, let us remember that it is not only the macrophages which are infected by the HIV virus, there are also other cells which are infected by the macrophages, like T lymphocytes, okay, T lymphocytes are also affected by the HIV virus, called as what? Retrovirus. It is a retrovirus. So HIV virus is attacking the normal cells such as what? Macrophages and T lymphocytes, and at the same time we also should remember it is also infecting or affecting the B cells also, okay, it is also affecting the B cells. Now let us come back here. If you are seeing now if you are seeing this part, okay, now you should keep in mind that it is a retrovirus. Let us remember that it is a retrovirus. Now it is infecting the animal cells, we are saying, okay, it is infecting what? The animal cells. Animal cells means what? Especially the human cells. But otherwise we already said that this virus is also there present in the monkeys, but it is not infecting there. Here the retrovirus is infecting the animal cells, means human cells. What are those human cells? Normal cells such as macrophages or T lymphocytes or B lymphocytes. Now let us take and understand now this animal cell is the macrophage, okay, macrophage. So now first, virus infects the normal cells. Virus infects the normal cells. Now when it infects the normal cell, here it is introducing, okay, it is introducing the RNA, okay, viral RNA, the complete virus. When it attaches to the animal cell, it is introducing what? The viral RNA. So viral RNA is introduced into the cell, okay, viral RNA is introduced into the cell now. So once this viral RNA is introduced into the cell, means what? It is this diagram represents the cell. So here the viral RNA, okay, the viral RNA is present where, means it is present in the cytoplasm of the cell. So remember, where this viral RNA is present, it is present in the cytoplasm of the cell, animal cell. What could be that animal cell? It is the macrophages. Now once it is introduced, okay, viral RNA is introduced into the cell cytoplasm. What happens to this particular viral RNA? A very important point we have to remember is that this viral RNA is having the ability to undergo reverse transcription to form as what? Viral DNA. Viral DNA. Which means if this is the RNA, okay, if this is the RNA molecule, this RNA molecule can be changed or replicated into a DNA molecule, This is RNA and this now we should remember it as DNA, okay, this is the DNA. How is this possible? Because of the presence of an enzyme called as reverse transcriptase, okay. There is one enzyme called as reverse transcriptase. Please remember, it's a very important enzyme. Very important enzyme we have to remember, which is involved in helping in formation of DNA from the RNA, okay, from the RNA. A single stranded RNA can replicate to form into a double stranded DNA because of the presence of what? Reverse transcriptase enzyme, So as soon as the viral RNA is introduced into the cell cytoplasm, this viral RNA is produced. Viral RNA will now, with the help of reverse transcriptase, it helps in formation of what? Viral DNA. Viral DNA is formed. So here, viral DNA is produced by reverse transcriptase. Reverse transcriptase is what? It is an enzyme. Please remember, it is an enzyme that is produced by the HIV virus, only. So this enzyme helps in converting or helps in replicating the viral RNA into DNA. Now what is produced in the animal cell cytoplasm? Viral DNA molecule is produced, okay, viral DNA molecule is produced. From where this viral DNA molecule is produced? It is from the viral RNA, okay, viral RNA. So viral DNA is produced by the enzyme called as reverse transcriptase. Now what happens to this viral DNA? Now viral DNA is formed. This viral DNA is second step, okay, second step. Once it is introduced, what is happening? It is forming as viral DNA. Second step, if you are seeing, this viral DNA, where it is present? We said it is in cytoplasm. It is incorporated into the host genome. Host genome means into the host genetic material. This is the cell and here is the nucleus, okay, here is the nucleus. So we know that inside the nucleus what will be there? The genome, genetic material, okay. Inside the nucleus what will be there? Genetic material will be there. Now what happens is that viral DNA that is formed is now incorporated into the host genome, okay, incorporated into the host genome. So let us make it very clear. Say suppose if this is the nucleus, okay, let us say that this is what? The nucleus. The same thing here, this nucleus, okay. If this is the genome, okay, this is the genome of the cell, okay, genome of the cell. Now this viral DNA is incorporated means here it cuts and this DNA comes and attaches here, okay, comes and attaches here, okay. It means it is forming as though it is the genetic material of the animal cell itself, okay, animal cell itself. So once it is incorporated into the host DNA, okay, host DNA or host genome, now what happens? Now that particular part of the DNA, viral DNA introduced into the host genome also undergoes the normal process, okay, the normal process called as transcription and translation, which you will be studying in the other chapter. What do you mean by transcription and translation? So here what happens is, because of that viral DNA responsible in producing a one particular protein, okay, or different types of protein for its okay, for its functioning, it is now introduced into the nucleus. And now here the third step what happens is that new viral RNA is produced by the infected cell. Now this is the cell, okay, this is the cell. Now once the cell is incorporated with the viral DNA, that viral DNA helps in formation of new viruses. That viral DNA present in the nucleus of the animal cell helps in what? Formation of new viruses that are produced. Now here this is the infected cell we are saying. Now if you are saying this is the genome, okay, of the virus that is newly formed from the viral DNA. Okay, from this viral DNA again what is formed? Viral RNA is formed. This viral RNA now starts synthesizing the core as the same here. Viral RNA core will be produced, and also the viral protein coat is also produced around it. So like this, there will be many viruses that will be formed. There are many viruses that are formed, and these viruses, they come outside. Okay, they come outside and they infect the other cells. What could be those other cells which will be infected by these new viruses that are synthesized in this animal cell? Now they may target now the T lymphocytes followed by the B lymphocytes. Now we know that the major cells that are very important in the immune system are what? T lymphocytes. So when the T lymphocytes are being destroyed, what happens? The immune system becomes very weak. The immune system becomes very weak. So here what is happening is, when the animal cells such as the macrophages are infected by the HIV virus, the viral RNA is multiplied or undergoing reverse transcription with the help of an enzyme called as reverse transcriptase, forming viral DNA, and this viral DNA which is present in the cytoplasm enters into the nucleus of the animal cell, and there it helps in formation of the new viruses, means new viral RNA. And this viral RNA, okay, forms as it starts forming all the parts that are required, forming the new viruses. These viruses are coming out and infecting the T lymphocytes. T lymphocytes are very important cells in the immune system, and once they are being destroyed, now the immune system becomes very weak. Okay, the immune system becomes very weak. So this is a repeated process, and remember one very important point, you have to make note of that. When this is taking place, okay, remember the animal cell is not getting destroyed. The animal cell is not getting destroyed, okay, until these viruses are formed, okay, and released, okay. And that's why we have to remember that this animal cell is functioning, okay, it is functioning as what? HIV factory. It is functioning as the HIV factory. Please remember this, okay. Animal cell, that is macrophages, are functioning as what? HIV factories. When these viruses are being formed, please make note that this animal cell will not get destroyed until the formation of many viruses, and these viruses are infecting the T lymphocytes, we said. And when these T lymphocytes are infected, we are telling that the immune system becomes weak, during which time our body or that particular person's body who is infected with HIV, okay, will be showing some symptoms, okay, will be showing some symptoms. What could be those symptoms? Bouts of fever, okay, bouts of fever. At a regular intervals, that particular person will be keep on, okay, suffering with fever, okay, that we are calling as bouts of fever. And he or she may also suffer with diarrhea, okay, he or she may also suffer with diarrhea, and slowly they lose their weight. So they, in HIV patients, okay, suffering with AIDS, what will be seen? Weight loss will be there, okay, weight loss will be there. And please make note, weight loss means it's not, we should not misunderstand it, that it is because of what? AIDS. There are many other ways or other reasons why a person will lose the weight. There are many other reasons why the person will get fever or suffer with diarrhea. But it should not be confused that if he or she is suffering with bouts of fever or diarrhea or the weight loss, it may be AIDS. But if these three are being at a time seen as the symptoms, then they have to cross check it, because these three at a time are caused because of what? The HIV virus, which is destroying which cells? When this HIV virus is destroying the T helper cells, okay, T helper cells. So when the T helper cells are getting destroyed, our immune system is becoming weak, and they show the symptoms like bouts of fever, diarrhea, and weight loss. And with these, we also should make note that due to decrease of T lymphocytes, they suffer from infections. Okay, they suffer from infections. Already we said, when the T lymphocytes are less in number, the immune system is becoming weak. When the immune system is becoming weak, there are chances that there are many microbes may enter inside into our body and cause their different diseases. So here, as soon as the T lymphocytes are being destroyed, okay, they may be suffering from different infections. What would be those infections? Especially bacterial diseases like mycobacterium, okay, or like some viruses or fungi, and even a parasite called as toxoplasma. Okay, there is also a parasite called as what? Toxoplasma. Okay, toxoplasma is a parasite. Okay, it is found in the soil and especially in the fecal matter of the cats. And when it is contaminated with the vegetables or with the fingers, and when a person eats the food containing this parasite, okay, single-celled parasite, toxoplasma, he or she may suffer with what? Headache, fever, okay, headache and fever and muscle pains, okay, headache, fever and muscle pains are caused because of this particular toxoplasma parasite. Maybe for a short period of time or for a long period of time, he or she may suffer. Okay, so these are all the diseases, okay, causing microbes which can easily infect a person when their immune system is becoming weak. Now the question is, when the immune system is becoming weak, when the T lymphocytes are being decreased in number or destroyed. How these T lymphocytes are being destroyed? It is because of the HIV virus which is destroying these T lymphocytes. So you have to remember, HIV virus, when it destroys the T lymphocytes, there are more chances for that particular person to suffer with different types of diseases, maybe caused by bacteria or viruses or fungi or any other parasites. Okay, these are some points that we have to keep in mind. Okay, we have to keep in mind. So now, how to identify that he or she is suffering with AIDS? There are different diagnoses, okay, that can be done for identifying the patients who are suffering with AIDS, or test. What is the test that can be done for confirming AIDS? Okay, what is the test that can be done for confirming AIDS? A very important test or diagnosis that is done for AIDS, to confirm AIDS, is the first test is called as ELISA. Okay, first test is called as ELISA. Please remember this. ELISA means enzyme linked immunosorbent assay. Enzyme linked immunosorbent assay. This is the test name. Okay, ELISA is the test name. Okay, it is called as what? Enzyme. Okay, enzyme linked. Okay, enzyme linked immunosorbent assay. Immunosorbent assay, we call it as immunosorbent assay. Okay, immunosorbent assay. This is the name of the test that can be the name of the test that can be done for, okay, knowing, identifying or AIDS. Okay, identifying the disease AIDS. There is another very important confirmatory test, very important. How do you confirm? This is a test, but how do you confirm that it is exactly AIDS? That is the second one. It is called as the Western blotting technique. Okay, the second one is called as Western blotting technique. Please remember this. This is the main test through which they confirm that he or she is suffering with what? AIDS. Please remember, confirmatory test. Okay, it is completely confirmed that he or she is suffering with what? AIDS by Western blotting technique. Otherwise, there is also another test or diagnosis that can be done for AIDS. Okay, that is called as what? PCR. Okay, PCR. Please remember this also. PCR. PCR means, okay, polymerase chain reaction. Okay, polymerase chain reaction. Okay, polymerase chain reaction. This is another, okay, test that is done to confirm the person, to confirm that the person is suffering with AIDS. So these are the points that we have to keep in mind, and this is a very important. Okay, this diagrammatic okay representation is very important for your subjective point. Okay, for exam point of view. So please keep these points in your mind, and we will continue to discuss about the AIDS in the next video. Thank you so much.